What is RNAi? RNA interference. It is a gene slicing mechanism where a double-stranded RNA leads to degradation of specific messenger RNA, and this prevents the production of specific protein. So here there is a double-stranded RNA. The double-stranded RNA can enter the cell naturally. Example, viral infection or artificially by research therapy. Sometimes this short hairpin RNA or the hairpin shaped RNA mimics dsRNA. It is also known as this short hairpin RNA is known as shRNA. Then there is a dicer that cleaves the dsRNA. An enzyme called dicer. It cuts the long dsRNA into short fragments called the siRNA, small interfering RNA, which is approximately 21 to 23 nucleotide long. Then siRNA is a double-stranded with overhangs. Then what is this RISC complex formation? One stranded of siRNA called a guide strand, it is loaded in this RISC, that is the RNA induced slicing complex, RISC. So the other strand, that the passenger strand, is degraded. So an important protein in RISC is AGO, that is Argonaute, which help to bind and cut the mRNA. There is target mRNA recognition. The siRNA within the RISC RISC binds to the complementary mRNA based on the base pairing, and this binding is highly specific, like a lock and key. Then mRNA cleavage, that is also known as the gene slicing. Once bound, this AGO slices the mRNA. This mRNA is then degraded by the nuclease, which results into effective slicing of gene, and no protein is made. So the key component of this RNAi mechanism is dsRNA, and its function is to trigger the RNAi. Then there is a dicer which helps in the cutting the dsRNA into siRNA. Then siRNA guide the RISC to target the mRNA, and this RISC the protein complex that degrade the mRNA. Then AGO is endonuclease that cut the targeted mRNA. So RNAi is use the siRNA to guide the RISC complex to specific mRNA and degrade it, and stopping the protein production. So how to remember? So let's make a mnemonic. Dicer, siRNA, RISC, target and slicing. For this we have make a mnemonic. You can imagine a chef, that is a dicer, in a kitchen, cutting here. What cutting means? Cleaving something raw. This raw tomato, it is cutting, the chef is cutting this tomato, that means dicer is cutting the RNA. Cutting means cleaving the RNA and making a small dish. Now the chef is cutting the tomato and making a dish, that means dicer is cutting the RNA and making siRNA and putting it on the plate, that is the plate is the RISC. So the dicer is cutting the RNA and making a small dish siRNA and putting it on a RISC, and then throwing away the spoiled ingredient. Now what it is throwing away? For example, the chef is peeling the tomato or peeling the potato, so the peeled ingredient, which is spoiled ingredient, which is not used in cooking, that is the mRNA. So dicer is cutting the RNA and making a small dish siRNA and putting it on a plate like RISC, and then throwing away the mRNA. Thanks for watching. Do like, share and subscribe.